following Bob is always the MIT version of freshman hazing. So most of you can tell I'm joined up here by my senior colleague, David Housman, who knows a lot more about all of which I'm going to talk about. So you can refer any questions to either of us. My lab is interested in cancer therapy, and in particular in why cancer therapies fail. And I think a good example of the problem that we work on is shown here. These are two lymphomas that are visually indistinguishable, and not only are they indistinguishable to you and me, but they're also pathologically indistinguishable. And because they're pathologically indistinguishable, they're treated the same way. Unfortunately, these two tumors have very different responses to therapy. This tumor responds very well, and this tumor shows no response whatsoever. So this patient gets subjected to the same chemotherapy and has the same side effects of being treated, yet this tumor shows no response whatsoever. And so the question is, what is the difference between this tumor and this tumor? What makes this tumor resistant to chemotherapy? Well, the difference is genetics, that there are mutations that promote drug resistance, just as there are mutations that promote cancer development, as Bob just discussed. And so if we could just identify the mutations that are present in tumor B that aren't present in tumor A, we could easily identify which tumor is going to respond to therapy. Unfortunately, that turns out to be a very difficult process, and that's because tumors are very complex genetic organisms. This is a spectral karyotype of a normal cell showing these matched pairs of 23 chromosomes, and this is a karyotype from a cancer cell, which, as you can see, is extraordinarily complex. There are many copies of many chromosomes, so you can have 8 or 9 copies of the same chromosome. And not only are there more copies of these chromosomes are rearranged and portions are amplified and portions are deleted. So there may be hundreds of changes in this cancer cell that are not present in this normal cell. And just looking at it, it's very difficult to tell which of these alterations were relevant for tumor development, which of them are going to impact therapeutic outcome, and which in the end are going to represent meaningful drug targets. And so I thought, because we are joining engineers, I'd give you an engineering example of this. It's a lot like comparing these two bridges, where this bridge to the right has a lot of problems. But most of these problems occurred after the bridge fell down, or as a consequence of this bridge falling down. And it's very difficult to tell just by looking at this what were the critical flaws in this bridge here that caused it to fall down in the first place. And so, like any good engineers, to get at this question, we try to model this process. And an example of this modeling is shown here. These are 3 mice, all of which have GFP-positive lymphomas. So you can see these green spots throughout these mice. And at day 0, all of these tumors look somewhat comparable. These 3 mice are treated with the same therapy, which in this case is cyclophosphamide, and they have very different responses to this same therapy. So this top tumor goes away, yet these bottom 2 tumors essentially stay put, or in fact they may grow a little bit. At about a week after therapy, both of these top 2 tumors are gone, yet this bottom tumor is still present. And at 2 weeks following therapy, we start to see this middle tumor relapse. And because they respond differently to therapy, they also have very different therapeutic outcomes. So this mouse on top is actually cured by this therapy. This mouse relapses at 3 weeks and rapidly dies. And this mouse actually has a slow-growing tumor and manages to survive for about 2 months following therapy. Now, because we created these tumors, we know what distinguishes them. And in this case, it's just a single alteration that distinguishes these tumors. So this middle tumor is the same as this top tumor, except it's missing an important tumor suppressor, which is called P53. This bottom tumor is the same as this top tumor, except it's overexpressing an oncogene called BCL2. So a single genetic lesion can determine the response to an existing chemotherapeutic. It can determine whether a tumor responds successfully, or a patient responds successfully, or a patient fails therapy. And so my lab is interested in examining this process, looking at many different genes in the genomes, and in fact performing screens looking for the role of thousands of genes in therapeutic response. So essentially our strategy is to start with tumor cells and introduce via retroviral infection either single genes or dozens of genes or hundreds of genes at a time, and look at the impact of those individual genes on overall therapeutic response. And so I thought I'd show one example of this process, looking at the frontline chemotherapeutic adriamycin. Can I get some water? Sorry. So adriamycin is a frontline chemotherapeutic that's used for a wide variety of epithelial cancers, including breast cancer and colon cancer. Is also used for, It's also used for a wide variety of hematopoietic cancers like lymphomas and leukemias. And so we performed a screen looking for the key determinants of response to adriamycin. And the gene that we pulled out as most responsible for the response of tumors to this drug was a gene encoding this protein topoisomerase 2. So what's a topoisomerase? Well, a topoisomerase is a protein that resolves DNA knots. So if DNA gets concatenated or knotted, then you can't have proper DNA replication, you can't have proper DNA segregation, so cell division doesn't work. So our cells rely on topoisomerases like topoisomerase 2 and another topoisomerase 1 to resolve these knots to allow cell division to continue. And topoisomerase 2 resolves these knots using a very specific strategy. It binds DNA at the site of a knot, and it makes a cut next to the knot. It sort of cheats in resolving the knot. It then resolves the knot and glues the DNA back together. Now, what we and other groups have found is that doxorubicin actually binds topoisomerase 2, and it changes the function of this enzyme. So it allows topoisomerase 2 to cut DNA. It just doesn't allow it to stick it back together. And so in the presence of doxorubicin, you have topoisomerase 2 cutting DNA throughout the genome, making lots of double-stranded DNA breaks, yet not resolving those breaks. So it's a topoisomerase 2 poison that makes a good enzyme a bad enzyme. And so the net effect of this is that cells that actually don't have topoisomerase 2 are resistant to doxorubicin, because doxorubicin can no longer bind this enzyme and promote lots and lots of DNA damage. So if we return to the initial lymphomas that I talked about at the beginning, we can start ascribing them a molecular signature in addition to this initial visual diagnosis. So say, for example, lymphoma A has topoisomerase 2, then one would expect that this tumor would respond to doxorubicin, whereas perhaps lymphoma B has no topoisomerase 2. And so in this case, doxorubicin treatment will fail. So you'll know going in the case of this patient, to give them doxorubicin and not even to try doxorubicin in patient B. Well, what do you do in this kind of situation where you have a patient that you know is not going to respond to a given drug? Well, as I mentioned earlier, topoisomerases are essential proteins. You have to untie knots that spontaneously occur in DNA. So you have to have topoisomerase 1 or topoisomerase 2. So if a cell is missing topoisomerase 2, what we can do is we can actually give these tumors a poison for topoisomerase 1. the net effect of this is eliminating all topoisomerase or unknotting capability in a given cell. So in this case, a cell dies because it can no longer carry on cell division. And so returning to our tumors, in this case, a tumor that has topoisomerase 2, we treat this tumor with doxorubicin. This tumor that has no topoisomerase 2 is now sensitized to the effects of another topoisomerase inhibitor. So in this case, we can treat these tumors with camptothecin. So this is exactly what we mean when we talk about personalized therapeutics, that we actually match up a given chemotherapy with a specific constellation of molecular genetics that are present in a given tumor cell, that we want to give the right set of chemotherapy to the right tumor. Importantly, as Bob Weinberg just mentioned, MIT for the last 30 or 35 years has done research to understand the molecular signature of a wide variety of tumors. We know a great deal about the molecular genetics that leads to tumor formation, and I think now is the point at which we can start to apply that information towards therapeutics, where we can think about what a particular lesion or genetic abnormality is going to do in response to a particular alteration. And in fact, I think we also know that alterations that are typically associated with drug resistance can now be regarded as tumor vulnerabilities. So any oncogene that actually promoted tumor development, I believe in some way is going to represent a lesion that can be targeted by alternative therapies. And again, we now have adequate understanding of molecular biology of a lot of these tumors that we can start to apply this information towards tumor therapies. And so I'll end there and answer any questions, or David can.
Mike, let me ask you a question to get you started.
Yep.
There's a lot of interest now in genomic approaches to cancer and genome resequencing. It's going to give more information about genes that are mutated in a given tumor, at least a class of tumors. Can you address that new information from the perspective of personalized therapy?
Well, I think that there's tremendous value in understanding what makes a specific tumor specific, what makes it unique. And so to the extent that we can characterize a set of alterations that are going to be specific to a class of patients and correlate that with a clinical response, we're going to significantly accelerate our ability to personalize therapies for those patients. Whether that will require lots and lots and lots of tumor sequencing, I don't know. I think that there are lots of other approaches that we can use, like screening approaches, that can lead us towards critical nodes that are going to determine therapeutic outcome as well.
So Robert's asking whether the diagnostic approaches that are now coming to the fore, will they be of use in matching tumor to a therapeutic regimen? Well, since I asked the question, I mean, no question. That's one of the things I think to pay a fair amount of attention to, in the sense that in the laboratory, some of the information that we're concerned with in terms of bringing therapy accurately together with tumor are well established, and we follow these things closely. Bringing those things into the clinical setting in a useful and meaningful way is something that will happen, but is always a challenging enterprise, because you really have to collect very large data sets and really come to very clear understanding of the meaning of those data sets and particularly with respect to treatment outcomes, which take time. So, this is going to happen. It's something I think that will be interesting to follow and write about, because the landscape of cancer treatment will change in the next few years. Even as you do get a diagnosis, you'll no longer simply hear about your path specimen reveals these characteristics of your tumor. You'll get more and more information about the specific molecular characteristics of the tumor that the physician is going to attempt to treat. That is going to change, has started to change, but it's still early days, I think, Robert, with respect to that change in the clinical setting.
Yeah, I think that one of the exciting things about the work that a lot of the biological engineers that we're going to join in the new Koch Institute is a lot of them are focused on the kinds of technology that are geared towards understanding or following early detection of tumors, biological assays that can look at treatment efficacy. And so I think that the combination of looking at tumor genetics and being able to assay treatment response and having the technology to assay treatment response is going to be incredibly valuable.
Yeah, I could expand. Oh, great, we got a question. It's my old friend.
The slide you showed with the scrambled DNA from the cancer cell versus the normal cell, a huge difference in even the amount of DNA. Might that be an exploitable difference that you could look at as a way of attacking cancer, just because the amount of DNA or the energy needed to do the division or something like that?
Absolutely, and I think that there are certainly a number of groups that think about aneuploidy as a targetable condition. I think that there is a misconception among many people that cancers are robust systems, that they are hardier systems than normal cells. In fact, they're not. And carrying around that kind of karyotype comes at a cost. It's almost impossible to carry around that karyotype stably. So information would be lost with every cell division. Cytokinesis is likely a lot more difficult. And I think that the strategy, an excellent strategy, would be to try to essentially push that over the edge. And it goes back to this idea of tumor strengths, which are aneuploidy and the ability to introduce lots of additional alterations or changes in copy number, can also be tumor weaknesses if they're properly exploited.
Just to expand on that, the way in which, for example, that lymphoma lost the capacity to, the one that didn't respond to the adriamycin, lost the capacity to express the topoisomerase II, could well be a consequence of this loss of genetic information as the tumor scrambles its DNA in an attempt to adapt to the environment it is in. So there's certainly the possibility to tailor therapies to the scrambled genomes of the tumors. Good to see you.
So I actually have an additional comment about that, and then I'll ask another question.
Sure.
So you guys aren't asking questions. I'll ask. So Angelica Hemann, one of our colleagues in the Koch Institute, who's actually probably the most accomplished yeast cell biologist studying the cell cycle. She studies the cell cycle, how cells divide in a model system, yeast. And she specifically addressed this question of aneuploidy. Is there a price to pay for having too much DNA? And she discovered, using the powerful genetics of yeast, that there is. Yeast cells that have too many chromosomes are not fit. And then she's moved on to the mouse on her way towards humans. And the same thing is true. The mouse cells that have too much DNA are not fit. And so Michael's comment is right. There's a price to pay for that genetic scrambling. And maybe her hope is that can be exploited. So my question relates to therapy resistance again, from the point of view of the new therapies. You guys writing about the new era of anti-cancer drugs, Herceptin, Tarceva, Gleevec, wonder drugs, and they are wonderful drugs, but almost invariably, the patients relapse. And in some instances, the patients have no longer survival times than if they didn't take that drug. And that's the case for Tarceva, for example. So my question to you guys is, what are we going to do about that? The drugs work, but then they fail. So what are we going to do about that?
So I think that one of the challenges is understanding mechanisms of relapse and even predicting mechanisms of relapse, so that we can actually provide that in the context of initial therapies. If there are very common strategies that cells have for overcoming a given targeted therapeutic, then I think the idea would be to simultaneously provide a drug or concurrently provide a drug that's going to counteract that therapy. So in the case of EGFR mutations or BCR-ABL mutations in CML, there are drugs that can target a mutated protein. And so I think that you may want to preempt the acquisition of resistance by providing patients with drugs that are going to attack that resistant state.
Let me also add, part of the problem is we don't have enough of the wonder drugs yet. The answer, both for childhood leukemia, for HIV, is combinations. A single drug alone often runs into exactly the problem that we're seeing at this point with the Gleevec, Tarceva, and Herceptin stories. With a larger arsenal, the possibility of choosing the right combinations and essentially using our models to understand how to choose those right combinations is the way I think we'll get to essentially a final solution to the problem of these vexing cancers.
My question, maybe Professor Housman can answer this. I've written a lot about companies like Helicos that are developing DNA sequencers that may be able to achieve the $1,000 genome within a couple of years. And there's been a lot of hype about that, but I wonder how you, as researchers, envision the $1,000 genome accelerating your research into the clinic. Will it have a major effect?
First, a little bit of a caution about the $1,000 genome, because today you can go to the web, send a cheek swab off to any number of companies. 23andMe is the one whose name comes to mind, but there are a number. Send in your cheek swab and get back information. Whether that information is truly going to be useful to you in terms of your health and welfare is really the question on the table. So the first side of the answer to your question is, of course it will have tremendous impact to be able to gather information of the type that we've been discussing, so far this morning at a dramatically reduced cost, and by the way, perhaps an increase in accuracy for certain kinds of information we're looking for. That said, that's really only half the problem. The problem that really needs to be solved is interpreting that information and matching that information to the clinical situations that we're attempting to address, and really coming up with really correct and deep answers to what's really going on. That's really where the action will lie.
I mean, you think that even when you think about the sequencing of the previous sequence of the human genome sequence, and the promise that held and still holds for understanding complex diseases, I think that promise is still out there and largely unachieved in looking at polygenic diseases, heart disease, a variety of other conditions. I think that we have to think about cancer in the same way, that it, in many cases is going to be the complex synergy between a variety of different lesions that is going to provide a state that needs to be treated as a particular state, and is going to require a variety of different therapeutic strategies that's going to target a constellation of changes that are very specific. Our ability to pull out that constellation of changes from a much larger, diverse set of alterations is a real challenge, and I think that it involves a wide array of researchers, and it involves a lot of modeling of particular conditions by those of us that do that.
You had a follow-up?
I would say from my perspective, I think part of your question is how is that new technology going to enable us in the research setting? I think, one, it's going to be revolutionized. It's going to be yet another example of revolutionary advance, because right now, all of us are limited in the depth with which we can understand genetic information. And genome sequencing at that scale, that cost, gives us much more information. These guys are right. The information is only the beginning of the process. But we do need the information. And it's now going to be accessible in a very different way.
And it fits with the demos. One of the demos this afternoon will involve our high sequencing capabilities. You'll see exactly how it's being used in real time experiments.
Yeah, the new building will have an expanded suite of, we call them core facilities. It's not very interesting. It's actually 20,000 square feet of the new building will have a biotechnology center. And a big component of that is for genomic analysis, including DNA sequencing. It will not be as big a focus as it is in the building we're sitting in. The Broad Institute is an institute really devoted to genomic medicine. Their capabilities in the genomics space are greater and will be greater than we will be. Nevertheless, because it's a core component of biological research today, institutes such as ours need. So we have invested already in the
Yeah.
Other questions? Yeah. Which technology? So the question is, which technology have we selected? For the moment, we have placed our chips on Celesta, which is now owned by Illumina. But as your question alluded to, it's very rapidly changing, right? So whether that technology will be, and I'll get to David's point of view on this too, whether that technology will be the favored one in 6 months, I wouldn't want to say.
I can't add anything to that, Tyler, because I think we're constantly evaluating these technologies because they're critical. If the ability to gather information changes in terms of, essentially dollar per unit of information with any of these technologies, or if the accuracy of one becomes attractive relative to another, we'll be choosing that technology very rapidly, I think.
I think we certainly all benefit from our affiliation with the Broad, and most of us, I think, are formal Broad affiliates, and they certainly have taken a lead in the field, and so it's a real advantage having them guide us in those directions and provide a capability that really adds on to what we can do in-house.
How many of you have ever actually sequenced DNA? Anybody here in the audience? No, none of you guys? Yeah, one. So, do you ever actually have to distinguish whether a site was mutated in an individual or not, or something like that? No. So that turns out to be a harder thing to do with the old technology, seeing whether you have an A and a G together, whereas the newer technology now looks at individual molecules. So the molecule that has the A is sequenced over here, the molecule that's sequenced that has the G is sequenced over here. And that actually improves the accuracy for the critical thing that we're often looking for, which is the difference in one of the two copies, but not in the other. So the new technology has a value beyond its throughput. It's also very helpful in terms of finding the very things that we're most concerned with looking for.
And also in tumors that are very heterogeneous, being able to detect rare populations or rare mutations within a larger tumor that may point to an evolving mechanism of drug resistance or modulator of metastasis